Hello everyone, thank you for inviting me to talk at the Symposium series. I'm Rachel Hale. I'm a researcher at Cardiff University, but today I'm going to be talking about a project that I worked on at the University of the West of England in Bristol in the UK from 2015 to 2017 with Professor Julie Kent, who's now retired, Professor Maria Fannin at the University of Bristol, and Dr. Darian Meacham, who's now at the University of Maastricht. And we're hoping to publish further from the project, so any comments or questions would be welcome. So the study investigated attitudes towards an emerging technology, which is variously termed cultured or manufactured red blood cells or synthetic blood to treat patients with rare blood groups, sickle cell disease and thalassemia. And the targeting of red blood cell products to address putative unmet need is a focus for continuing research into red blood cell manufacture. And I'll consider what kind of future for blood services in the UK and cultured red blood cells are envisaged by scientists, patients, blood donors and publics. And particularly relevant for this symposium is whether the donation of peripheral blood or bodily tissues as starting materials for the culturing of red blood cells is an extension of the gift relationship of donation. So first I'm going to situate our research within the sociology of expectations and the future of regenerative medicine. Then I'll present our case study of synthetic blood or cultured red blood cells. And finally, I'm going to suggest that innovation in this area draws heavily on the institutional significance and the role of the current blood service provider NHSBT in the UK, particularly in England. So Professor Nick Brown and others have discussed the colonisation of the future, the way in which innovation in this area has been hyped by the media, government, clinics and companies is part of what may be seen as investing in the future by building hopes and expectations. And in an earlier paper with Mike Michaels in 2003, they discussed the fetishisation of the soon-to-be, the commodification of the future, but also how this generates greater uncertainty. The growth in knowledge, they suggest, is linked to more uncertainty for people, for example, about risks and safety of new technologies. The construct of the breakthrough in scientific narrative has been constant and enduring, but they argue this misrepresents a process of experiment and clinical translation. Often futures fail, including for some regenerative medicine. So building expectations and a future for emergent technologies like cultured red blood cells is a key aspect of contemporary bioeconomies. The creation of new networks, infrastructures and institutions are important aspects of designing futures. Promissory narratives work to create and produce futures that generate interest, investment and potentiality. I'm now going to situate the development of cultured red blood cells in the current UK blood economy. So around 3% of the UK population are blood donors. There is an ageing donor population and concerns about the recruitment and retention of future donors. There are 4 UK blood services, NHSBT in England, and 3 smaller organisations in Scotland, Wales and Northern Ireland. Also, there are 6 public cord blood donation sites in the UK, and these are set up in areas where banks are targeting diverse ethnic groups. And over 20,000 donations have been banked, making it the 4th largest cord blood bank worldwide, with the 2nd highest percentage of rare tissue types. And key users of blood products are patients with blood disorders such as sickle cell disease and thalassemia, patients having major trauma, major surgery or childbirth. As I said earlier, there is putative unmet need for some blood products from people with rare blood groups and with some blood conditions that require regular blood transfusions, and cultured red blood cells are being designed as a solution to these issues. I'll say more about the claims being made about cultured red blood cells shortly. First, I'll say a bit more about the science of this technology. So cultured red blood cells can be grown in the laboratory outside the body from a number of different sources of stem cells, namely donated human peripheral blood, umbilical cord blood, embryonic tissue, skin cells, and bone marrow. Different research teams are using different cells to culture red blood cells. So in Edinburgh in the UK, they are using human embryonic stem cells and induced pluripotent stem cells. And in Bristol in the UK, they are using stem cells from donated peripheral blood, umbilical cord blood, and growing immortalized cell lines. And there are different ethical issues with the different starting materials. The first in-human clinical trial, which is used in healthy volunteers, is called RESTORE, which stands for Recover and Survival of Stem Cell Originated Red Cells. And the trials were due to begin in the mid-2010s in the UK. And the most recent update on the webpage says that this has been pushed back to 2021. And I recently requested an update and was told that the trial has started, but they cannot give any updates on the trial as it has not gone into the public domain as yet. I'm now going to talk about our case study. So the National Institute of Health Research Blood and Transplant Research Units in the UK are research partnerships between universities and NHSBT. Excuse me. We work with Bristol Blood and Transplant Research Unit to achieve one of their objectives, which is to engage with patients and publics to determine the acceptability and attitude towards cellular therapies as an alternative to donor-derived red cells. So the aim of our study was to investigate how blood donors, patients, publics and key stakeholders think about blood donation, the existing arrangements for collection and distribution of blood, and also the development of cultured red blood cells. We conducted a qualitative study using focus group discussions and key stakeholder interviews with a total of 43 participants. We conducted 2 focus groups with blood disorder patients, including thalassemia, sickle cell disease, myeloma, essential thrombocythemia, and myelodysplastic syndrome, and 1 individual interview with a haemophilia patient who wasn't available for the focus groups. We also conducted 2 focus groups with blood donors, one with mothers of babies, and we were particularly interested in their views on umbilical cord blood donation, and also 2 focus groups with other members of the public. We also conducted 8 key stakeholder interviews. 3 were from UK Blood Services, including 1 cultured red blood cell scientist, 2 were doctors, and 1 was a nurse from haematology services, and 1 was a transfusion specialist, and we also had 1 patient group representative. We also carried out a literature review, including NHS, Bristol BTRU, Edinburgh University Novosan and Rosalind Cell's websites. I'm now going to talk about the institutional arrangements. From the review of websites and previous literature. Excuse me, my slides seem to have skipped ahead. So I'm now going to talk about the institutional arrangements for the development of cultured red blood cells in the UK. So there are 2 research groups which are developing cultured red blood cell technology. In England, it's led by NHSBT and Bristol BTRU, and they are using donated peripheral blood and cord blood, and have developed the first immortalized red blood cell called Bella. And as one of our interviewees said, they were not happy that the name is so similar to Hale. In Scotland, it's a consortium led by the Scottish National Blood Service. Also involved are several universities, Cell Therapy Catapult and the Irish Blood Transfusion Service, and they're using human embryonic stem cells and induced pluripotent stem cells. So far, this research has been publicly funded with significant public investment from NHSBT, NIHR and the Wellcome Trust in these research groups. And in order to take the research forward in the future, the research groups need to attract private investment. and this has been done in Scotland through two spin-out companies. So Novasang is a future-oriented entity created to help build expectations and futures for cultured red blood cells, mainly by attracting investments. And Roslin cells have generated human embryonic stem cells to clinical GMP grade. So from the review of websites and previous literature, we identified several claims around the future of cultured red blood cells. So these were around addressing UK unmet clinical need, so more precise matching for rare blood groups, inherited blood disorders, other hemoglobinopathy patients, and to reduce the effects of sensitization and iron loading in regularly transfused patients. The second claim was around addressing shortages in the international blood supply general populations. Then as a viable alternative to large-scale blood donation through industrial-scale production, as a safer alternative to conventional blood donation through manufacturing standards, to reduce costs of importing blood products derived from stem cells, and for human enhancement in terms of much improved physical ability such as speed, strength and stamina. I'm now going to consider some of the promissory claims about cultured red blood cells in the context of the data that we generated with our research participants. So the role of NHSBT emerged as central because they have the existing infrastructure for collection and distribution of blood products, and they have relevant experience in manufacturing facilities. Some key stakeholders seem to see NHSBT's involvement in developing this technology as a step in their involvement in other service areas, particularly regenerative medicine, rather than as purely an interest in cultured red blood cells. And one blood services participant described cultured red blood cells as low-hanging fruit because the blood services already have the expertise and infrastructure for handling red blood cells, and they also know what this product, a red blood cell, should look like. This participant proposed that the primary driver for developing red blood cells is regenerative medicine, and that the field is booming at the moment, and that as an organisation, NHSBT wants to be involved in that in terms of the lead therapeutic that they can develop based on their expertise. Their unique position is a manufactured red blood cell. Indeed, one of NHSBT's strategic goals is to strengthen their position in the development, assessment and clinical delivery of regenerative medicine-based therapies, and cultured red blood cells are the only technology mentioned on their website regarding this goal. And as for the public view, we found considerable trust in NHSBT blood services amongst our focus group participants, and confidence that if NHSBT was involved, then this would assure the product was safe, properly tested and well regulated. And a blood donor focus group participant said, "At the moment I trust the NHS to be as good as its word, so I would like to have knowledge that it's through the NHS and through some cabinet's best friends company out in Liechtenstein who's doing this. In other words, the commercial bits. It's a great idea, as with the stem cells, but you can imagine that people would want to exploit it. So if it's commercially based, then I get suspicious. But if it's good for the group of people under the NHS banner, then I'll be happy." As I said earlier, cultured red blood cells are being developed to meet putative unmet need for cultured red blood cells. However, some stakeholders propose that there is in fact decreasing demand for red blood cells due to changing practices to reduce the use of blood overall. And this was identified as a threat to the financial viability of existing blood service organisations. And there was no consensus among stakeholders about whether red blood cell demand will continue to increase or decrease, and stakeholders were less able to see a future for cultured red blood cells among the general population as a universal product. There was also a difference of opinion between a patient group representative and a transfusion specialist as to whether cultured red blood cells as a niche product could better meet any of their patient needs. In addition, one stakeholder proposed that scientists developing this technology might have motivations less to do with translation into the clinic and more for the development of science per se, and that discussion of the science is competing with getting cultured red blood cells as the market. Focus group participants also envisage cultured red blood cells having a role in developing other technologies, such as growing genetically modified blood or entire organs in the future, and a key stakeholder proposed that they might be used as a diagnostic tool. So there are lots of claims being made around cultured red blood cells, but our stakeholders revealed many aspects of uncertainty around the development of this new technology. And given the early stage of development of cultured red blood cells technology, this is perhaps unsurprising, and these uncertainties didn't necessarily detract from support of the concept of culturing red blood cells or enthusiasm for their potential therapeutic application. While the focus group's participants may have had less knowledge about the blood economy and cultured red blood cells than the key stakeholder interview participants, this difference did not reveal itself in much concern about uncertainties around cultured red blood cells. And as has been suggested in previous research about the uncertainty trough, expert scientists are more likely to be uncertain about the future, but also capable of being entrepreneurial and making strong knowledge claims. The uncertainty about cultured red blood cells mainly came from our stakeholder participants then, and we've grouped these under 4 areas. So wider aspects of the blood economy in terms of supplies of donor blood, demand for certain blood products, and target markets for cultured red blood cells. Scientific questions about culturing cells, testing them in humans, and assessing outcomes, and aspects of the manufacturing process and ensuring a safe blood product. And then finally, translation of the technology to the clinic, including funding available. As well as uncertainties, participants also identified other future challenges for cultured red blood cells. So there's competition from other research groups and also other therapies for some of the target patient groups. There are external risks from other research groups with public awareness of issues around cultured red blood cells, even if from other countries, could by association negatively impact on investment and social acceptability in the UK. There is also the challenge to develop the expertise needed to translate the technology to the clinic. There are financial and reputational risks to the blood services involved in developing the technology and risks to donor relations if they mistakenly believe that they are no longer needed to donate. And reconfiguration of blood services will need to consider the impact on current and future employees. I'd also like to highlight that our participants' concerns are in contrast with Mitra et al.'s paper, which focuses on the regulatory challenges of cultured red blood cells. So our study has illuminated the promissory narratives around this technology. While there seems to be support among both publics and stakeholders for cultured red blood cells, our participants also discuss many of the challenges and uncertainties that will be faced in translating this technology to the clinic. And while the public seems to be primed to accept NHSBT's reconfiguration as a regenerative medicine supplier, could the public be expecting too much? And how will NHSBT be affected if they cannot fulfil these expectations? Furthermore, in the reconfiguration of blood services to accommodate cultured red blood cells, there is also a threat to donor relations and retention. On the one hand, NHSBT wants to promote the idea that cultured red blood cells are an innovative technology that has potential to satisfy unmet clinical needs, but they also want to retain blood donors and the blood supply and the idea that it is business as usual. And finally, our clinicians and patients also revealed that there will be work to to be done in diffusing the technology to the clinic. And this picks up on themes identified by the Regenerable Med project in its 2017 policy briefing on the clinical delivery of regenerative medicine. We have a list of references available for anybody who would like them if you want to contact me. And we would also like to thank all our organisations and individual participants that took part in the research and helped us to recruit participants as well. Thank you.